So we also just discuss to use the yeast as the host to express the protein, right? And plant, animal, all right? So this is the same insect system. And then after you produce the protein, then the next step is to purify the protein, all right? The protein purification, basically the first step is to prepare an extract from the purification. So basically you harvest the cell and then you need to break the cell, all right? You need to break the cell because cell has a cell wall, so you disrupt the cell wall and release the protein. That's the extract. So once you have the extract, then you can perform the protein purification. Okay, so that's the first step, and to collect the cell extract, and then you need to stabilize the protein in the solution, and because the protein can be degraded very quickly, so you have to always maintain the low temperature and the proper pH in the buffer, all right? And sometimes you have to add the protease inhibitors and the antimicrobial to prevent the protein to be degraded. And also you add the additives to prevent the foaming and the shearing from destroying the protein. So that's how you stabilize the protein in the solution. And then you prepare the separate component in the extract, all right? You separate this component in the extract. You have these several approach that we would discuss, okay, later on. And then this is one of the methods that separate the component in the extract by doing the protein precipitation, okay, because the protein is hydrophilic, a lot of hydrophilic amino acids on the surface, so you can add the salt and mix, and then to precipitate the protein, so you can separate the protein from the from other component. And the most important technique is to use the chromatography to separate the protein from other components in the cell extract, and which is the one we're going to discuss here. Okay. So basically, the most important technique people use to purify the protein is called chromatography. All right. So the chromatography, basically, you have two phases. One's mobile phase, one's the stable phase. The mobile phase, which will be the cell extract, and then you run through this the beads, glass beads, and the beads can be in the column, and then the glass beads, the beads column can act as the filtration system to separate the protein from the other component in the cell extract. Okay, so that would be, this is a general situation for the column chromatography you use to separate the protein. So you have a column, and then you add your sample to the column, and then because these beads, you have different property of beads, which we will discuss in the next slide. Because of this different property of beads, you can act as the filtration the beads can help to separate the target protein from the other component, depend on the property of the protein. Okay, and then you can elute the protein in the end. So that this is a very commonly used technique, and usually four different commonly used column chromatography methods: ion exchange, reverse phase, gel filtration, and affinity. Okay, and so for the ion exchange, it depends on the property of the beads, all right? So that's here is the 4 different general property of the beads that you can use. So for example, the beads, if the ion exchange, all right, you separate the protein by the electrostatic interaction. So the beads can be a positive charged or the negative charged, all right, depend on the protein that you want to purify. So if it's cation exchanges, cation beads, they can separate the positively charged protein. For anion exchange, separate the negative charged protein. For the reverse phase, so basically you use the different concentration of the salt, so you can induce the protein to be the hydrophobic, and then you can do the hydrophobic interaction chromatography, which is the experiment that we need to go through today through the video. All right, that's the hydrophobic interaction chromatography. That's reverse phase, and then you use a high salt concentration, and then you change it to the low salt, and so that you can purify the protein. And geofiltration, that based on the size, the molecular weight of the protein, that you can separate the protein. Affinity, so that specific ligand binding, and commonly seen affinity chromatography would be the antibody of chromatography. Okay, so the first one here, the size exclusion, that will be the geofiltration, right? Based on the molecular size, that's the size exclusion. The size exclusion basically that all the beads here, all right, the beads, they have different size of sieve based on the molecular weight cut off molecular weight. So for example, if you say the molecular weight cut off molecular weight is the 60 kilodalton, so that means all the protein, all right, larger than 60 kilodalton will be excluded from the beads, and anything smaller than 60 kilodalton, so smaller than the cut off, then they will be trapped by the beads. All right. So when you have the cell extract to go through this geofiltration beads, all right, so those protein larger than the cut-off size will be excluded. Then they will go through the column quickly, and then you will obtain the protein first. So you see the large protein first, and then all the proteins smaller than the cut-off, they will be trapped by the beads, and therefore they'll be eluted at a later time. All right, so you will get the large protein first and then small protein later. So that's called size exclusion chromatography. And this is just an example to show the low molecular weight, high molecular weight. High molecular weight will go through the column first, and then the low molecular weight protein will be trapped in the column, and then they will go through this slowly. All right, so this is the size exclusion chromatography. And then ion exchange chromatography that rely on the electrostatic charge to bind protein to the resin beads in the column. Okay, and so when the charged protein bind to the resin, then the contaminant will pass through and out of the column. All right, so an ion exchange resin is positive charge. All right, and then cation exchange resin is negative charge. So cation exchange resin will bind the positive charge protein, and ion exchange resin, because it's a positive charge, so they will bind the negative charge protein. All right. And then here's the example, right? So in this example, so basically when you run through this column, right? Run through this column, then So for example, in this case, you have the positive charge ion exchange beads. They will bind the negative charge protein. All right, bind the negative charge protein. So the anion exchange resin is positive charge. All right. And so they bind the negative charge protein. So when you run through the column, so the most negative charge protein will be eluted the last. All right, and then the positive charged protein will be eluted first. All right, so that's the principle of the ion exchange chromatography. Okay, so this is in the low salt concentration, because this is positive charged beads, they will bind the anion negative charged protein. All right, when you increase the salt, then finally those negative charged protein will be eluted. Okay, in the beginning, in the low salt condition, all the negative charged protein will be bound to the beads, and then other protein will be excluded. And later on, you slowly increase the salt concentration, and then you will elute those negative charged protein. All right, and finally the high salt concentration. Then all the negative charged protein will be eluted. Okay, this is an ion exchange chromatography. Any questions so far? Good. And then the affinity chromatography. So rely on the ability of most protein to bind specifically and reversibly to ligand, right? And the most commonly seen example will be the antibody, the protein bind to the antibody, okay? So the affinity ligand, the column resin beads that has the affinity ligand. So when you run through the cell X-ray, the protein will bind to the ligand. And later on, you use a high concentration the protein, the peptide, to elute the target protein. So that's the affinity chromatography. And then the reversible chromatography. So the example is hydrophobic interaction chromatography. Okay, the protein are salty on basis of their repulsion of water, and the column beads are coated with hydrophobic molecule, and then the hydrophobic amino acid in the protein are attracted to the similar chemical in the beads. So basically, you need to treat the protein with the high salt concentration solution, so that the protein will make those hydrophobic amino acid outside of the protein. Therefore, when you go through the chromatography column, then those protein will bind to the beads. And later on, then you reduce the salt concentration. Then those hydrophilic amino acids will stick out again, and then will bury those hydrophobic amino acids, and then you can elute your target protein. So that's a hydrophobic interaction chromatography. Okay. So basically, the two different type of resin. All right. So you have active substitute resin and the phenyl substitute resin. Okay. They can interact with the nonpolar amino acid side chain of protein, and then the protein can be released by lowering the salt concentration, low salt concentration. Okay, so this is just an example. So when the protein you treat it high salt, all right, and then all those hydrophobic amino acids will stick out, and then they will bind to the resin. And later on, when you lower the salt concentration, then they will bury those hydrophobic amino acids, and then you can elute those protein. So that's a hydrophobic interaction, chromatography. So we are supposed to show this video again. Let me try one more time.